Hello again, everyone. In this video, I'd like to discuss insertion and deletion mutations and how they have an impact on gene expression. So this is video 2 of a little mini series on mutations. There's another video that covers substitution mutations and the biochemistry of a certain substitution mutation. So if you're looking for that, check out that video. So here I will just discuss insertions and deletions. But first, I'd like to remind everyone about the molecule that's actually getting mutated. So DNA gets mutated. A lot of times students will refer to RNA or proteins as mutated. It all starts upstream at the DNA molecule, so keep that in mind. Each cell has its own copy of the genome, which is all the genes a certain species will have in its cells. So each cell only expresses a subset of these, but each cell has that full copy. This copy acts as a blueprint for the construction of all these proteins that the cells will need to be able to perform all of its functions. So when that DNA gets mutated, you see the ripple effects in RNA and in protein, but it's that first upstream molecule of DNA in the nucleus that gets mutated. The importance of this can be seen in what I call the cake pan analogy. So remember that gene expression takes DNA and converts it into molecules of RNA through a process called transcription. Okay, and that RNA is converted into protein through the process of translation. If you have this DNA molecule, you get this RNA, and from that you get this protein. So we're well versed in gene expression. If we were to convert this over to a baking analogy, if you have square cakes or square cake pans, you make square cakes, will eventually lead to square birthday cakes. Okay. However, if you drop the cake after baking it in a square pan, you'll have a problem. You wouldn't want to take this on to make a birthday cake out of it, but you could always go back to that square cake pan and make another one if that was the problem. However, when we're talking about DNA mutations, the mutation lies in this analogy in the cake pan. So if you drop the cake pan, all cakes that come out of it will have the same indentation. You never want to take those to the next level in this analogy of making a birthday cake. But if we're talking about molecules like DNA and RNA, if you have DNA here and your RNA is the molecule that gets mutated, you can always go back to DNA and make a copy of new RNA. But because DNA is the molecule that gets mutated, every RNA molecule made from it also harbors that mutation. And if you were to go on and make proteins, that change would be reflected in the protein as well. So just a quick analogy to help follow the flow of the mutation and the consequence it has, first on RNA and second on protein. So let's take a look at the consequences of various mutations in DNA using some simple 3-letter English words. So in my mind, this is an excellent way of illustrating the impact that insertions and deletions have on DNA structure and ultimately protein structure and function. So it's a nice general perspective. So if you have this statement, the dog ate the cat hat, although it's a weird statement, you definitely get the message just by reading that. So you get imagery of a dog eating a cat hat. Okay, so if we were to illustrate various mutations, let's first start with a substitution mutation, just as a quick review. If you were to change just one letter, you can see in this illustration, everything else has been the same in this code, if you will, but we've had a substitution here where there should have been an H, now I've substituted a B. Everything before this makes sense and in fact is the same. However, this last little substitution changes a little bit of the story, but you get a lot of the story still. So while you do have that change, you're getting most of the message here to begin with. So it's not a good thing, but as you'll see in a minute, it's not as devastating as insertions and deletions. So let's look at an insertion of another letter in this sentence. Okay, so here if you insert even one letter, especially early in the message, let's insert an A here. Everything else downstream of that T is going to push down. So here you'd have the H next to it, but this E pushes down. So E, D, O. Okay, so your message goes from being the dog ate the cat hat to ta ito gat ethe aka. So you can see that's pretty much gibberish. The same can be seen inside substitutions. Or I'm sorry, deletions. So let's look at a deletion. Let's instead delete this H right here. So then the message becomes okay, so it's an equal amount of gibberish, and that is because you're seeing what we call a frame shift. So the frame of the three-letter words has shifted such that now they're not in the correct place for them to be understandable. So basically it's become unintelligible gibberish. And the same can be said for your DNA code, as we'll see in a minute. If you shift the frame of ultimately your RNA codons, then the amino acids that are produced in translation also all change. Okay, so the consequences for this are absolutely devastating. Hopefully you can see that from that illustration. Okay, so let's look at this in DNA sequences. On the left you have what we call the normal DNA code. It is the most abundant version in nature, so organisms that are in a population of whatever organism this is, if you look at a certain sequence of their DNA, they would probably have this sequence of DNA. Not that there aren't other sequences, but we're calling this wild type. Okay, so you can see the wild type DNA on the left, wild type RNA on the left, and then ultimately wild type amino acids for this segment of the protein. Okay, so you follow it through, and for wild type, the amino acids that you should have are isoleucine, glycine, isoleucine, glycine, lysine, and leucine. Okay, and then very importantly, a stop. However, if we insert a T into this genetic material right here, as seen here, everything shifts down, and ultimately you get different codons. So you can see, just glancing from wild type to the mutant on the right, due to the insertion, all the codons have shifted. Okay, so when you have this frame shift, a lot of bad stuff starts to happen downstream, and you can see it play out when you start to translate. So if I translate the codons that were produced, now instead of having an isoleucine here, I have an asparagine, a glycine, and importantly, I have an early stop. So this UAA stop codon comes prematurely and produces a stop, which makes the protein smaller than it should be. So ultimately, you get different amino acids added to the protein. If you recall in the previous video, we call this a missense effect. That video goes over, comparing various amino acids and the biochemistry of a substitution. So if you want, check that out. In addition, you see the introduction of an early stop, or what we call a nonsense effect. Okay, so it's important to designate those two different outcomes from, in this case, this insertion mutation. We can also see this play out in a deletion. So again, if I were to take this A and delete it as shown over here, everything in this case then shifts up. Okay, so you can see that that A at position 2 has been removed. All of the letters ultimately in the RNA shift up and your codons change. You can see just from the very beginning, AUC over here, ACG over here. And what you're going to see is a long run of missense changes. And eventually, in this case, instead of having this, you have all of these totally different amino acids. And here, where we should have had a stop, here, this is going to continue on until the code runs into a stop. So in this case, you'll have a bunch of missense changes, and the protein will be longer than it should have been, which is also a problem for its function because we have lost the stop there. Okay, so just a quick overview rundown of insertions and mutations, insertion and deletion mutations that should complement the previous video on substitutions. Okay, I hope everybody is well, and until next time, we'll see you then.